Hello everyone, we are sending our best wishes from the team in Leicester. I hope each and every one of you have been keeping safe and healthy. For those of you that don't know me, my name is Mervyn. I'm a clinical lecturer based at Leicester. This means 50% of the time I'm doing clinical work and the remaining time I get to do exciting research in the field of nystagmus. Although it's a shame that we are not able to meet in person, we are glad to see Nystagmus Network organizing this event, and really it is our pleasure to be part of the 2020 Nystagmus virtual event. So together with my colleagues Helen, Rebecca, Irene and Frank, we would like to share an update of our work at Leicester. I first thought it would be good to give you an overview of the research team based at Leicester. Many of you will have come across our lovely research orthoptists. In fact, some of them are very experienced, even completing PhDs in the field of nystagmus. Viral is one of our most experienced orthoptists who is also a postdoc researcher in our group. Similarly, Gail also completed a PhD with our group but has moved now to the University of Sheffield as a lecturer. But she still remains an active part of our research studies and remains an honorary fellow with our group. Helen is currently completing her PhD, and Michael has been with us a number of years doing research and clinical orthoptics with our group. Many of you already know Rebecca McLean. She's been with our group for over 20 years. She now leads the clinical service and the eye movement service at the University of Leicester. Frank Proudlock also doesn't need any introduction, an experienced veteran in the field of nystagmus with over 20 years experience. He's an associate professor with a background in engineering and neuroscience. Tu Zanan is also a postdoctoral researcher, did a number of studies on electrophysiology of the retina in patients with nystagmus. The clinical doctors in our group include Professor Irene Gottlob, who many of you would have come across during your clinic appointments. Myself, Mervyn Thomas, I'm a clinical lecturer. Dr. Sohaib Rufai, who is a doctoral research fellow, and Dr. Arti Patel. So she's just completing her doctoral research fellowship at the moment. It's a tough task to summarize all the nystagmus research that goes on in Leicester, but over the next few minutes I aim to give you an overview. Overall, our work is divided into specific themes, and these themes include basic science research into nystagmus. These include novel gene discovery, molecular biology studies trying to understand on a molecular level how does nystagmus occur. Improving diagnosis. A large part of our clinical research is driven by trying to improve diagnosis in children with nystagmus. This includes pioneering genetic studies, use of handheld OCT. We were the first center in Europe to introduce this, and also using more recently artificial intelligence in the field of nystagmus. We also are quite interested in psychophysical studies and nystagmus. This includes face recognition and nystagmus, the quality of life in nystagmus, and how do individuals with nystagmus read. In terms of treatment, I will also give you an update on some of our exciting surgical outcomes in patients with nystagmus and abnormal head posture. Finally, I will touch on some interesting research on the prognosis in patients with nystagmus. So we ask a question: can we predict future vision in young infants, and young children with infantile nystagmus? First, I will touch on some of our genetics work and how we are aiming to improve diagnoses in nystagmus. It is helpful to reflect back on the genetic studies carried out at Leicester, but overall what I'd like to show you is how far we've come in understanding nystagmus genetics, the heterogeneity of this disorder, so the diversity of the disorder, and how we've changed the way we test and look for genes and mutations in individuals with nystagmus. So our story begins back in 2006, where we identified one of the major causes of infantile nystagmus, and this was known as the FRMD7 gene. This formed the stimulus for a large part of our genetic and clinical studies. This was followed on by a number of interesting papers where we characterize this particular condition in a large amount of detail and also show another condition called periodic alternating nystagmus, where the nystagmus changes direction over time and the amplitude also changes over time. This all culminated in 2011 into the first NHS genetics test for infantile nystagmus, and this was a collaboration between the team in Leicester and Nottingham, thus giving patients for the first time back in 2011 access to a genetics test on the NHS. From there, we did a number of molecular biology studies and also looked at other conditions that can cause nystagmus, including the PAX6 gene. This is a gene that's known to cause loss of iris tissue, known as aniridia. But what we found was there is such a variability in the way this condition can present that some individuals can just have nystagmus, underdevelopment of the retina, they can have early cataracts, but no iris tissue loss. So maybe some cases were missed previously, and that was some of the basis of some of these studies that we presented. We then went on to look at how does the retina differ in these people that used to be called idiopathic nystagmus. And idiopathic nystagmus used to be thought to be arising from a problem more centrally in the brain rather than from the retina. But what we found was we looked at the expression, which means where is this gene expressed? And we found strong levels of expression within the retina. And this is what formed the basis of could the neural center that's causing this be retinal? And that's what we've now come to realize more recently. This then led on to in 2015 for the 100,000 Genomes project. So many of you may have heard about this. This is a big initiative by Genomics England, and essentially they wanted England to be right at the forefront of genomics research. But we were initially disappointed because nystagmus was not part of this 100,000 Genomes project. So we championed to bring forward nystagmus as one of the conditions that should be studied as well under the 100,000 Genomes project. Thus, it gave patients for the first time access to something known as whole genome sequencing, where the whole genome is sequenced, not just one gene. And so patients now, at that time, I guess, would have had access to the 100,000 Genomes project as we nominated nystagmus to be studied there as well. Simultaneously, what we were doing was we were working on something known as a nystagmus gene panel, and this is to show proof of concept on the clinical utility and validity of capturing multiple genes at a time. So if you remember back in 2011, we only had capabilities to sequence or to look at one gene at a time. But we've come a long way now to the point where now we show that actually we can target multiple genes simultaneously, looking for molecular errors that might be causing nystagmus. And as many of you might be aware, nystagmus can arise from mutations in genes in lots of different genes. So it's helpful to do this in parallel rather than doing one gene at a time. Subsequently, what we did is we had a number of multi-center collaborations, and in 2019 we identified another nystagmus gene, and this gene was called AHR. At the last meeting, at the last workshop, we had discussed this in more detail. And more recently, we've been also characterizing other nystagmus genes that have been poorly characterized in the literature. And in fact, this one gene known as the SLC38A8, it can mimic a lot of the features that we see in albinism. So often, patients who have mutations of this gene are often misdiagnosed. Therefore, in this particular study that we completed just last year, we give an algorithm that helps clinicians to help diagnose this condition that also can have underdevelopment of the retina and poor cone photoreceptors. So these are what are responsible for your 6/6 vision or high area of visual acuity. And one exciting area of research that we recently started is the Nystagmus twin study. And the Nystagmus twin study looks at patients who have identical genotype, so they have the same genetic makeup, but could they have differences in the way nystagmus presents? And that's a really exciting area of research, because this now helps us provide information as to how much of nystagmus develops from the genetic makeup compared to environmental influences. One of the biggest advantages of the nystagmus gene panel is that we can test simultaneously for many genes at a time. Therefore, we advocated for an improved diagnostic workflow to improve the diagnosis. What this means is that it can be used as a frontline diagnostic test, either using just saliva samples or blood samples. We can obtain DNA and test for all the known genes associated with nystagmus. This may well obviate the need for some of the other investigations. But there is still additional work that is needed within this field, because right now we still face a significant lag time between obtaining a sample and providing the results. But over time, as the technology improves, I anticipate we will be able to reduce this lag time to obtaining results, and therefore it can be actionable results in real time. Sticking to the theme of improving genetic diagnosis, using our nystagmus gene panel, what we found was that a specific group of patients were often being misdiagnosed, and these are individuals that had SLC38A8 mutations. To extend our study further, we performed a multi-center collaborative study with our colleagues in Korea and also other parts of UK. And we found that actually 90% of these patients were initially misdiagnosed as having albinism, for instance. Moreover, we also characterized the retinal development using a technique called optical coherence tomography, which obtains higher resolution images at the back of the eye. We also characterized the nystagmus waveforms for the first time in this group of patients. Based on our findings in our previous multi-center study, what we realized was it would be quite helpful for clinicians to have a diagnostic algorithm. So we published this specifically to assist clinicians who might be faced in similar scenarios with infantile nystagmus, so they can then decide what specific genetic subtype they could be faced with based on things like your retinal imaging scan, the optical coherence tomography. The Nystagmus twin study is an exciting project currently underway at the University of Leicester. In theory, identical twins share the same genetic makeup, thus any differences in the clinical characteristics can be attributed to other factors such as environmental influences. We hope that this will therefore decipher the contribution of genes and environment to the development of nystagmus and other clinical characteristics such as the underdevelopment of the retina. If you or a family member are interested in supporting some of this research and are an identical twin with nystagmus, please feel free to contact us through Nystagmus Network. Some of you have already enrolled in our study, and we are so grateful for your contribution so far. Now we will briefly touch on some studies looking at improving diagnosis in nystagmus using specialized retinal imaging techniques. Some of you may have come across a machine called optical coherence tomography, which helps obtain high resolution retinal scans. It gives unprecedented level of detail of the development of the retina. On the left, there's a table mounted machine that you may come across at opticians and a number of hospitals, which is often used in imaging, especially in adults. However, this is not accessible for imaging in children. Sometimes they use a method called the flying baby position to try and acquire retinal photos in children. This is the image shown on the right. However, as you can imagine, this is not the most elegant method. This is where the use of a handheld machine has been particularly useful in infants and young children, especially with nystagmus. At Leicester, we were the first center in Europe to pioneer and advocate its use for pediatric ophthalmology and in particular for nystagmus. We are now pleased to see more centers have followed suit and have incorporated this into their clinical practice. This is a video demonstrating the use of their handheld OCT in a young child. And my colleague here is Dr. Arti Patel. She first sort of demonstrates it on herself, makes the child comfortable, and then you will notice that the probe comes quite close to the eye, but it actually doesn't contact the eye, so it doesn't touch the eye. So this is a non-contact, non-invasive method of acquiring images very fast of the back of the eye. You can see within a few seconds you're able to acquire images of the retina and the optic nerve. Now we will switch gears slightly and focus a bit more on some of our artificial intelligence projects that we are currently undertaking. At the last meeting, we had the privilege to meet some of you and share some of our preliminary results on using artificial intelligence to differentiate between normal and abnormal retinal development. This has significant impact both for diagnosis and prognosis. And we achieved very high levels of accuracy, 96% accuracy, in terms of being able to do that on a binary scale, meaning between normal and abnormal. So over the last year, we kept building the system further, and this was to improve the clinical utility of this intelligent diagnostic system. What we wanted to do was to further subclassify the abnormal types of retinal development to provide more diagnostic and prognostic information for clinicians. And this also we were able to achieve 95% diagnostic accuracy. The other step that we carried out was also aiming to make our system device agnostic. What this means, it doesn't matter where you have your scan done or on what device. It would be completely separate device, for example, even a table mounted device at your optician, but the intelligent system would be able to cope with that. So this work has really been showing some proof of concept for potential as a frontline diagnostic tool in patients with abnormal retinal development, which is often seen in individuals with nystagmus. Now we will touch on some psychophysical studies that have been carried out in Leicester. This is an example of a video showing an overlay of where the eyes are moving whilst reading a paragraph of text. What we find is that normally individuals would have these quick eye movements called saccades, followed by a brief pause, which is the fixations at different words. Now, in nystagmus individuals, we find that they read at a slower rate compared to controls, and this is because of the nystagmus being superimposed on the normal reading pattern. However, in some rare instances, what we did find was the quick phase, which is the quick eye movements in nystagmus, had been suppressed, and they utilized the slow movements of the eyes to glide the eyes right across the text and acquire the information that's required. So this was an adaptive strategy which we rarely did encounter in some individuals. Another area of interest is facial recognition, quality of life, and emotional perception in individuals with nystagmus. This work is really spearheaded by Rebecca and has produced some seminal publications in this field. In this recent study that is ongoing, that Rebecca is carrying out, what they're looking at is facial recognition in particular. So recognition of faces and correctly reacting to emotional changes in facial features is extremely important for social interaction. And our previous research has suggested that people with nystagmus struggle with facial recognition and social interaction. So the study, what we were proposing was to assess whether people with nystagmus do underperform in facial recognition tasks and what are the reasons if this was the case. In this series of studies, we utilized standardized computer-generated facial expressions and looked at facial recall in a time-restricted environment, visual search in a crowded search field, and emotion recognition. This was looking at differences between individuals with nystagmus and those without. This is ongoing work, so you will have to wait. So unfortunately, I will keep you in suspense till the next meeting for an update on the findings. Now I will briefly hand over to Frank to just update you on some of our surgical outcome work for treatments in nystagmus.
Hello everyone, this is Frank Proudlock from the Leicester group, and I'd like to spend a few minutes explaining some of our results from looking at the effect of head posture surgery. It's work that has been carried out by one of my colleagues, Swati, and the surgeries have been primarily done by Irene Gottlob. And what Swati has done is what we call a retrospective study. That is to say that she's gone back and she's brought together all of the data that we've collected over the years, and she's brought it all together and she's analysed it. The main results are shown in this quite complicated slide here, so I'll just spend a few moments explaining what we've got. We've got the effect of the horizontal head postures surgery on the left hand side, and then vertical head posture surgery on the right at the top, and then the torsional head posture, which is this kind of posture, if you like, if you can see the video, is at the bottom on the right hand side. And really what the graphs are showing is the amount of head posture before surgery is shown by the blue squares, and then immediately after surgery by the yellow circles, and then the final head posture by the crosses as well. So, to cut a long story short, from the point of view of reducing head posture, the surgery is very effective. In most cases, the head postures are really brought down to either no head posture at all or a very small head posture, and it doesn't matter whether that is horizontal, vertical or torsional surgery. We've also looked at the visual acuity data. That is how much improvement you get in terms of getting down the vision chart where the letters get smaller, that you've probably all done. And what we find with visual acuity is that it doesn't make a huge amount of difference. Although there is an improvement overall, those improvements are usually quite small. When we first looked at the data, we thought there were certain patients that were responding quite well to the surgery and were really improving in vision quite a lot. But when we looked at the data more closely, we found that they were mostly children, and we think that their vision would have been improving anyway. So that's the story, if you like. Head posture surgery works, but it's good at reducing the head posture. It doesn't have a major effect on improving vision. I hope you're all doing well, and best wishes to you all. I hope your meeting goes very well. Thanks very much.
The last aspect I want to touch on in this presentation is regarding the prognosis in individuals with infantile nystagmus. At Leicester, we devised a grading system to grade the degree of retinal development from a scan. So these are scans that you have seen before using the handheld machine or a table-mounted machine. Based on the degree of retinal development, you can see that in the schematic shown here, the top panel, the figure shows a fully developed retina. So this has a deep pit, which is called a fovea. Fovea is just a Latin term for a pit. But then as you go through the different grades, for example, grade 1, there's a rudimentary pit. In grade 2, there's no pit. And each of these panels show loss of specialization, or these unique features that are present in the retina. Now the question we ask now is, can we predict the future vision in a child that comes to our clinic with nystagmus? So based on a scan in a preverbal child, before they can articulate or we can get an accurate vision, can we look at the retinal development and say, this is what their future vision is going to be? So this particular study required a lot of patience, as it was a longitudinal study, meaning that we had to call back individuals, specifically small children with infantile nystagmus, and see how their vision develops, but also look at their original scans when they first came to our clinics, when they were preverbal, so we couldn't get a reliable vision for that matter. So what we found was actually that the Leicester foveal hyperplasia grading system does indeed predict their future vision based on their initial scan. So this has powerful implications, because a child that comes to clinic now, we're able to do a quick non-invasive scan and say, based on your retinal development, this is what we anticipate the vision is going to be. So to conclude, really, we've tried to give you a flavor of the different types of research in the various themes that we have within Leicester. This is in improving the diagnosis and pathways within nystagmus, psychophysical studies, reading, face recognition, emotion recognition, treatments, specifically surgical outcomes, and anomalous head posture correction in infantile nystagmus, and more recently, really powerful data showing that we can predict future vision in children with nystagmus. So it's really finally just to say a big thank you to all our collaborators, our funders, and above all, none of this would be possible if it was not for the willingness and patience of all our nystagmus volunteers and family. So a really big thank you from all of us in Leicester.